Thank you to Rick and Susan for inviting me to present our work on studying cellular responses in severe COVID-19. This really is not much that needs to be brought up about this in terms of the background of COVID-19, but certainly lots of different symptom issues, some severe, some not as severe, ultimately in some individuals resulting in death. We know there's a lot of comorbidities associated with disease severity. However, as we've heard today and from Scott as well, both Paul and Scott, that there are many people who have asymptomatic infections. And so when this started in the end of March, we wanted to really begin to characterize what happens during the acute phase of infection in these individuals, as everybody essentially is naive to this, and so it's a really a wonderful opportunity in an adult population or even in an elderly population to understand the acute phase kinetics of a new pathogen from the scientific aspect. What we do know is that those people who have recovered do have antibodies, as Scott has shown us, and there's some reports out there that show that these individuals also have T cells in the blood. But of course, during the acute phase, there's a lot of questions as to what happens and what drives severity. And so the questions that we had at the beginning of this study were really, are these immune responses helpful or harmful during these early phases? One thing we'd really love to know and are continuing to look for samples are what are responses like in asymptomatic people, and there have been some reports coming out of this recently, especially from the Karolinska group of Marcus Bogert. What are the right immune responses to have? So what would you want to have coming out of this to be protective in the long run? And then ultimately, of course, a lot of work in terms of what therapeutics or drugs might work against SARS-CoV-2. At Penn, we started working, John and I started collaborating very early in this process and coordinating with the hospital to try to get access as early as we could to a number of different patient groups. And really the star of this is Nuala Meyer, and she was able to get us connected into the ICU where she was seeing many severely infected individuals with ARDS or the need for supplemental oxygen and so on. And so we started to coordinate a collection of cohorts with Nuala, and this evolved over time and started to increase in the number of different studies, and I've listed just some of them here. ICU patients with severe disease, non-ICU patients with moderate disease. John Wary has been collecting convalescent donor samples for some time. Scott just spoke about the healthcare worker seroprevalence study. I believe in the next talk, Ed Behrens will talk about the pediatric cohorts that Laura Vella and he and others at CHOP have been working on. Katie Barr, of course, and Don Siegel have been working on convalescent plasma therapy. And then there's a host of clinical trials. I don't pretend to know all of them, but John Wary would be able to give anybody information as to the different ones out there. And what we set out to do really was to take more of a step back from what was in the literature and really just ask the general questions as to what is going on during these severe infections. The data that I'll show you today has turned into 2 manuscripts, both of which are available in bioarchives. And mostly I'll talk about this paper on the left side, which is just a very general capture of as many different lymphocytes in both innate and adaptive immune lineages as we could capture in a relatively easy way. This study was led by Leticia Curry-Cervantes and Bettina Pampena in my lab. And then I will also touch upon work from John's lab that we all collaborated. Of course, we were all collaborating together on these that really focuses more on T and B cells. So I'll bring both of these papers into account as I present our work. Just very quickly about the MESSI-COVID study that Nuala Meyer has been running. And so this started off as the MESSI study, which was a study of sepsis in ICU patients. And it was modified to start to include COVID patients. This is the demographics of the MESSI subjects that are just the part of our paper that's referenced in the bottom. And so we have a group of mostly severe individuals, some with moderate infection and some with recovered infection. These definitions of severe and moderate were made clinically by Nuala. They were assessed based on primarily the need for supplemental oxygen or ventilator usage and so on. As is characteristic of the cohort in the demographics in Philadelphia, we had a high degree of Black or African American individuals that were in the cohort, and most of the severe infections were male, as we have come to learn. A host of comorbidities, including the sort of the classic things we've heard about: obesity, hypertension, cardiovascular disorders, type 2 diabetes, and so on. And even though when Nuala designated these as moderate or severe, based on the clinical needs of these individuals, when she went back and she used the Apache 3 score, which is a very complex definition of severity based on a number of different clinical parameters, moderate and severe cohorts very clearly were separated based on this independently determined Apache score as well. The Apache score also increased with age, so we of course know that the severe infections or severe outcomes tend to be more aged. That correlates with the Apache score. And as one would expect, based on severity, we have a host of different clinical parameters that were elevated in these severely infected individuals. For example, D-dimers are highly elevated, CRP, and so on. So that's just to give you a picture of the cohorts that we're working with. So we set out to study basically the immune landscape or the immune atlas of severe COVID-19. And we started off with this because the literature that was available at the time, and this was at the end of March, the beginning of April, was really rather piecemeal. A lot of papers at that point, and still continue to be found only on preprint servers, and most of the literature out there is on relatively small numbers of individuals, sometimes not fully defined in terms of severity. So we really wanted to step back and do this as best that we could. So we started off with using, rather than peripheral blood cells, which is what a lot of individuals' papers have been out there that are published, we set off using whole blood, and we used a very broad leukocyte lineage panel. And for anybody who wants to write this web address down, we have uploaded all of our flow data for anybody who registers on the website to be able to download and analyze these data for yourselves. And the data from the Dibbage Matthews paper is also available at the same site. In our work, we used fresh whole blood that was processed within 2 to 3 hours post-draw and stained, and we did this to capture the granulocyte and lymphocyte populations. And I already talked about the disease severity scale. The type of analysis that we did is shown here on the right. Scott's lab does ELISAs, my lab does flow cytometry. And so we use this extensive panel that is really, was actually designed for our type 1 diabetes work. We adapted that here to more fully capture granulocyte subsets and so on. And if you, since this is, again, the technical part of the seminar, here's some brute force flow cytometry data, basically just to demonstrate to you that we can capture with this panel really every major immune lineage of cells by at least a few markers uniformly across the board. And so we would start off a simple thing like CD3 positive T cells. We can identify both subsets. We can identify B cells, and including plasmablasts, which are the acute phase antibody producers that expand during acute infection. We can identify NK cells, monocytes, dendritic cells, immature granulocytes, innate lymphoid cells. We can divide the dendritic cells into plasmacytoid or conventional dendritic cells. We can subdivide the monocytes into classical, non-classical, intermediate. We can subset NK cells. Then within the T cell populations, and here just showing CD8s, We can subset into the different memory populations and so on. And then we can combine some of these markers to even identify additional populations of cells that might be even more somewhat cryptic or rare subsets of these. And so when you take all of these data, which can be very highly multidimensional, one way to depict them is to show what's called a T-SNE analysis. It doesn't really matter so much where these different colored blobs are on these plots. It's more really meant to focus on what's present on one side versus the other. So this is just showing one healthy individual and one severely infected individual. These are total CD45 positive cells. So these are total leukocytes. And what you see, as one would expect here, this humongous black blob are neutrophils. And so those typically overrepresent all your white blood cells in your blood in a healthy individual. And that's also the case in severe COVID-19. But what you can see is the relative size of some of these other populations changes in severe COVID-19. And I'm going to get into that for the remainder of the talk and focus on some of these. If we get rid of the granulocytes and look at lymphocytes, so this would be typically now what you might see in a lot of the manuscripts that are out there, you can start to see the differences in the different populations. So for example, here in the healthy, we have a population in red, which is nearly completely absent in the severe COVID-19. These are what we would call mate cells. We have the appearance of this purple population, which are B cell plasmablasts. These are the antibody producers. You almost rarely ever find them in a healthy person because they're only an acute phase response to an infection of some sort. And we see massive presence of these types of cells in severe COVID-19. I won't go, I'll go from here right into this plot, which shows every subset that we've measured. This is now showing the percent of total leukocytes, what are the most frequent populations or what populations are being lost. So if we start from the top, neutrophils become very prominent in severe infection. And I should say in all of these columns, we have healthy donors, we have the moderates, we have severe, and then we have individuals who have recovered from COVID-19. So during the severe phase, we see this heightened presence of neutrophils. We have an elevation of eosinophils as well. And conversely, we have a loss, actually, of lymphocyte populations. So B cells are down, T cells are down. Within T cells, both CD4s and CD8s, we have a loss of mate cells. We don't see a lot happening with monocytes. Other papers have found, that other studies have found that maybe there's an elevation in monocytes. We don't actually see that. We have a decrease in dendritic cells, innate lymphoid cells, and NK cells. And then we have this sort of appearance of immature granulocytes, which you can't appreciate here as being statistically significant, but other reports have shown that these are becoming elevated, and perhaps we see in some of our individuals an elevation as well. So sort of some of the hallmarks of this are this heightened neutrophil to lymphocyte ratio that has been touched upon in some preprint papers out there, and lymphopenia rather than leukocytosis. So during a viral infection, one would usually think you would have elevated white cell counts, B and T cells expanding, but we actually have the opposite. So going back to the clinical status of these individuals, you can take these Apache scores, so this metric of severity, and if you look at the percent of T cells, the percent of these mate cells, the percent of NK cells, the percent of B cells, all of these inversely correlate with the Apache score. So the higher the score, the lower these populations are. We also see this greatly heightened ratio of neutrophils to T cells and PMNs, and another another conglomerate of granulocytes, the PMN to lymphocyte ratio, both become severely elevated during severe infection. And these ratios also correlate very strongly with the Apache 3 score as well. So now I'll get into sort of some of the interesting things that we found during our exploration of these data. So I'll start first with the innate subsets, and then we'll go into the adaptive subsets. So here showing neutrophils and NK cells. In our panel, we identify neutrophils as being CD15 positive and CD16 positive. And so this is a healthy donor. And what we began to notice was that while we could still see neutrophils, and as I showed you before, they become greatly expanded in severe COVID, we were seeing this interesting loss of CD15 and CD16 expression on these cells. So they still express some of these markers, but their intensity is much lower. And this actually, we were seeing this on neutrophils. You can see it here also happening on NK cells. And if we do this type of t-SNE analysis where we now are focusing really on the brightness of CD16 MFI amongst all the lymphocyte populations, you see a lot more CD16 expressing cells. These are the neutrophils that become expanded in severe infection, but their intensity decreases. So the closer to red you get here, the more CD16 is on the cell surface. And in the severe infection, although we have elevated numbers of neutrophils, they're expressing on a per cell basis actually less CD16. And this was happening in the NK cells as well. We looked across multiple subsets at this same issue with CD16, and here plotting the neutrophils. Monocytes in some individuals, we were seeing overall low responses, but generally not a lot of CD16 expressing monocytes. The CD16 wasn't changing much. In K cells, we were consistently seeing this decrease. And on immature granulocytes, so these would be the precursors to neutrophils or other granulocyte populations that generally fall within a lymphocyte gate by flow cytometry. These are also can express CD16 normally, but are decreasing their expression. We worked on this a lot because we were really concerned that perhaps we could be wrong about this effect. But we went into the companion paper to our work done by Dibbage Matthew from the Wary lab. And this was an analysis by Bettina in my lab where she went back to Dibbage's data, which was all PBMC, not whole blood. And she selected out what would be these immature granulocytes and lymphocytes, and then just plotted this MFI of CD16 amongst these side scatter bright cells, which we're calling immature granulocytes, and the CD16 expressing lymphocytes, which should mostly be NK cells. And you can see very, very clearly that CD16 is downregulated on this completely independent group of analysis and data that was run simultaneously, but with many, many more donors. As you can see, it's a very consistent result of the decrease of CD16 expression. So we also wanted to look a little bit more into these NK cells in particular. So NK cells do decrease a little bit in terms of frequency in the severely infected individuals, but there's still some there. And we can see, again, I've already showed you this decrease of MFI of CD16, but we were concerned, well, are the NK cells dying? Are they changing in some other way? So we did a little bit of an extended separate panel analyzing various additional NK cell markers, including NKp46, NKp30, which are sort of classically defined identifiers for NK cell populations, NKG2A and NKG2C. And what you can see is these two receptors, NKG2A and NKG2C, which have activating or inhibitory functions. So NKG2C being an activating receptor, NKG2A being an inhibitory receptor. We see this elevation of NKG2C in the COVID individuals, and although not statistically significant, it appears as if there's also perhaps an increase in NKG2A on these NK cells also. So with that, I'll switch to the adaptive subsets and come into some additional striking findings that we had. One of them being this massive expansion of plasmablasts. So as I've said before, these are really sort of the precursors to plasma cells, which are what we think about as the long-lived antibody producers that you find typically in your bone marrow, but their blood stage are more what we would consider plasmablasts. And these are known to respond to be a response to an acute viral infection, for instance. So in a healthy donor, a 1.1% response of B cells being plasmablasts is almost sometimes even on the high side. Whereas in the severe COVID individuals, we were seeing some of the individuals with striking expansions of plasmablasts. One example here, and when we first saw this, we were concerned that this was a leukemic donor, but in fact, this individual is not leukemic. 96% of their B cells were plasmablasts at this peak severity stage. To put this into perspective as to how high this is, if you look at acute influenza and acute Ebola, where it is considered that these plasmablast responses can expand greatly. You can see that maybe in the case of Ebola, we're comparable, but these responses are well above what's been shown in flu, and nothing, at least in the literature, would approach this 96% number for any known viral infection in humans. Interestingly, one might think, well, is this a pathogenic effect? We did not find an association between the frequency of plasmablasts and the Apache score. So no association there. It doesn't matter how severely infected you are. You could still have this very highly elevated plasmablast response, even if your Apache score is not as high as others. We went on with the question. We worked with Nina Luning-Preck's lab to address these plasmablasts in a different way, and that question was: can we determine if these are likely to be SARS-CoV specific? We did not use at this point any types of probes yet to do this, but basically we took just peripheral blood B cells, which would include these expanded plasmablast populations, and we had Nina do B cell repertoire analysis. So what we're showing here is a mixture of severe, which would be all these individuals with an S on their at the beginning of their abbreviation, and moderates, and then we have a few healthy donors here as well. And what you're looking at here are fraction plots where you take the fraction of all the clones that you find in the B cell pool, and then how many of them, for example, would be composed of the top 10 clones, the top 11 to 100 clones, and so on. So the larger this yellow bar is, the fewer clones that you have, and the more expanded those clones are, is the best way to look at it. And so what you can see in these severely infected individuals, you have these and this is plotted in decreasing rank, basically. But you have this expansion of these of single or small numbers of large clones in these individuals, which you don't tend to see as much in these healthy donors. If you take the length of the CDR3 of these clones, you can see that the CDR3 length of these clones tends to become elevated in the severely infected, and this is shown here by looking at different clones and looking at the CDR3 length. You can see in the severely infected individuals, they have a much longer CDR3 length than do the healthy donors, implying some selective expansion of particular clones that seem to be looking for their antigen of some sort. So the question that we're going to be addressing in the future using B cell probes to pull out the SARS-CoV-2 specific is what can we show for the actual SARS-CoV-2 specific B cells? We work with Scott as well. He and his lab, I should say his lab, because Scott didn't do this. Scott's lab quantified the RBD-specific IgM and IgG responses in these individuals. And as you can see, even though these individuals are have severe symptoms, they're actually not having any problem, for the most part, making a very good antibody response. So these are falling right into, and some of them even higher than some of the plots that he showed in his talk for these severely infected individuals. And we were also able to see that this is days since onset of symptoms versus the level of antibody for the RBD domain. The longer these individuals were infected, the more likely they were naturally to acquire IgG of the RBD specificity. So this is data from Divige's paper to show the memory B cell populations. Now what we've done here is we're not looking at plasmablasts anymore. We're now asking what happens in the memory B cell populations. And so we see a decrease, not of naive B cells, but a decrease in these not switched or switched memory. So this is probably because of the expanding plasmablast population being taken out here. They did a Tijnia type of analysis to highlight the differences in the COVID-19 state versus recovered donors versus healthy donors. And all you really need to see here is how separate these overlay plots are. And some of that is because of this CD38 or CD27. CD38 is highly expressed on plasmablast cells. We have an appearance of these TBET positive B cells, which are typically an antiviral type of B cell that are showing up in the COVID-19 positive individuals. and using their flow SOM clustering algorithms, they were able to find different types of populations that were selectively found in the COVID-19, including this cluster 1 and cluster 5. Cluster 1 here being a TBET positive B cell being elevated in the COVID-19 positive cell, and this cluster 5, which would fall into a highly cycling, probably more plasmablast-like population here. So now on to T cells. Although the initial reports on this went back and forth a little bit about T cell activation, we were able to see a very clear increase in activation within CD4 T cells in the severely infected individuals, and here as identified by CD38, by Ki67, by the combination of HLA-DR and CD38, as well as PD-1. And this, if we focus in on memory CD4 subsets, we can see that the severely infected individuals across all of these memory CD4 subsets, you can find cycling CD4 T cells within each of those populations. Same can be said for CD8 cells, where we could see sometimes greatly elevated CD38 and HLA-DR CD38 positive cells within the severely infected individuals, sometimes the moderates as well. This extended again into all the different memory CD8 populations, including TEMRAs, central memories, effector memories, and the transitional memory cells. One important thing to note from this, however, is that not everybody has T cell activation. So this is data from Divige's paper with a much larger number of donors. And what you can see here is that although some individuals have this very coordinated CD4 and CD38 activation occurring at the same time, you actually see that many of the COVID-19 populations, patients, actually fall into these categories of having low levels of activation. So to take all of this together, I'm going to show 2 more data slides and then wrap it up. We took all of our data. This is from Letty and Bettina's paper. We took all from that one single flow panel. We were able to discern all of these different factors from just one flow panel. We were able to, in an unbiased way, take all these data and then put them into an unbiased algorithm. And as you can see here, the severely infected individuals very clearly cluster out. And if you put this into a PCA analysis, you can see, based on all of these parameters together, that you can see the severely infected individuals clearly segregating away from the healthy donors, and as well as the recovered individuals. The primary drivers of this tend actually to be a lot of the T cell activation, some of the B cell activation, as well as some of the CD16 issues. And if you start to look carefully through here, you can see that there are some distinct clusters here. These clusters became even more clear in Divige's analysis when he focused on B and T cells. And here they used the NIH ordinal scale to define severity, a scale of severity. And here just showing that as the darker maroon this becomes is the more severe part of the scale. And in Divige's analysis with Ki67, so CD4 and CD8 T cell activation as well as plasmablast expansion, the more severe these individuals became, the more likely that you were to see an elevation in these particular populations. And in their UMAP clustering, they were also able to find a distinct clustering of the severe cases away from the healthy donors as well as the recovered donors. This is just by looking at B and T cells. And I don't have time to get into this, but as they dug into this more deeply, they were actually able to find 3 distinct subsets of responses here, characterized by people who had high levels of T cell activation, intermediate levels of T cell activation, and no T cell activation as distinct clusters. So where are we now? So from our analysis as well as what was done by Divige, we know that innate immune lineage has identified perturbations that occur in severe COVID-19 disease across multiple immune subsets. So almost every subset that we've looked at, we can find things that are happening. So a massive marshaling of the immune response in these severely infected individuals. But are these a marker of immunopathology, and which ones could be targeted therapeutically to ameliorate disease? And which ones are associate long-term immunity, disease resolution? And why is there heterogeneity in the immune response? And will this necessitate tailored approaches to treat patients effectively? So some individuals may have heightened NK cell activity, others may have heightened T cell activity. Maybe the same types of treatments will not be as effective in each. Can we use these types of patterns to predict disease course? This is something we'd really like to do. So individuals who have moderate disease, will we be able to use these markers as a way to predict if they're going to go on to have a more of a severe course ahead of time? And how could we change these by therapeutic approaches? So I feel like I've gone over time a tiny bit. I'll go to my big, long thank you slide. A lot of people are involved in this. Here's my lab in the corner. Letty and Bettina really were the champions of all of this, and as well as the entire sort of first month's worth of COVID-19 sample processing at Penn. And Divage was amazing through this as well in John's lab. And then John's team really came in and did some massive amounts of analysis of all of these data, of Divage's data to find some things. Scott's lab helping out tremendously by doing ELISAs. None of this would have been possible without Nuala Meyer and her team